Hello, my name's Melanie Senior. I'm a biopharma and healthcare writer for Nature Biotechnology and Informer, based here in my home office in London. It's my really great pleasure to moderate the rest of this Reflections on the Pandemic session and to introduce and send a warm welcome to Professor Azra Ghani, who is Chair in Infectious Disease Epidemiology at Imperial College London. Now, Imperial College epidemiologists and disease modelers, like many across the world, have played a really important role in helping governments and policymakers to better understand COVID-19 and the virus that causes the disease. Professor Ghani herself is an expert in the dynamics of disease transmission and also explores the impact of different health policy interventions like lockdown, social distancing, etc., on the spread of disease. And she's worked on a range of different infectious conditions, including SARS, HIV, VSE, and we're frankly delighted to have her with us. What I've asked Professor Ghani to do in some prepared remarks is to just tell us how and when her team at the beginning of the year began to piece together the characteristics of the SARS-CoV-2 virus, its spread rate, its severity, etc., and then to describe how this information was shared with policymakers, but also addressing some of the challenges, the many challenges of tracking a very rapid outbreak involving a very new virus, and of course, of extrapolating data from one country, one region to the next, often fast evolving data. And then finally, Professor Ghani is an avid science communicator herself as well. She's going to tell us what she thinks has been the impact so far of the pandemic on the public's understanding of science and the scientific process, and also perhaps on its faith in science. So without any more from me, I'm going to pass the virtual podium over to Professor Ghani. And this is a live session, so please, those of you in the audience, do use the chat function at the bottom of your screens to submit any questions, because we are going to try and leave some time at the end for those. Thank you very much. Over to you, Professor. Thank you.
Okay, thank you very much, Melanie, and thank you to the organisers for inviting me to speak today. Hopefully in the next 20 minutes, I'm going to, as Melanie said, reflect on some of our experiences over this year in working on this coronavirus outbreak. So you'll all be aware that the SARS coronavirus emerged in Wuhan in China in late 2019 with early spread around a market. And global spread has been rapid, as we might expect for a respiratory pathogen. We now have over 11 million cases and 500,000 deaths worldwide. And the initial spread was really characterized by an early jump towards Europe, with epidemics occurring between February and April. And those have now declined due to strong control measures that were implemented. And now the foci has really moved to Latin America, to South Asia, in the Indian subcontinent, and also to a re-emergence of transmission occurring in many of the US states. So what does an epidemiologist do? Epidemiology is the study of the transmission of a disease, and we're aiming to understand how it's spreading, who the most vulnerable populations are, and what can be done to reduce the spread and minimize the associated health impacts. And one of the challenges for a new disease such as COVID-19 is that initially we know very little about any of these aspects. However, what we do know as epidemiologists is we have our vast experience of working with other similar respiratory viruses, and we know that spread can be rapid and the consequences could therefore be potentially high. So very much of our early work is trying to focus on understanding, is this a threat that we need to take seriously, and how far could it spread, and how severe could it be? So back in January, our initial aim was really to understand two things: how fast is the disease spreading, and how severe is the outcome in those who become infected. And of course, there is very little information early on, and we relied early January on case reports coming from the Wuhan area of China. And these were very patchy. It was very difficult to put together a clear picture, but some signals were clear even as early as the second week in January. And that's when we released our first report. And what we found then was we started to see cases emerging in other countries. And essentially, this is a bit of a canary in the mine sort of scenario, when we see those cases emerging, we understand what the travel patterns are. We have detailed information on flight data, for example, and we could already estimate that there had been around 1,700 cases by the second week of January in the Wuhan area. And what that indicated was that it was highly likely that there was person-to-person transmission going on. So moving forwards a little bit, the next step was really to try and understand how fast the virus is spreading. And this is the R number that we are now also familiar with, the average number of new infections generated by one primary infection. And if this number is greater than 1, then infection is spreading. And the relative speed of the spread will be determined by how much higher than 1 it is. And using epidemic data from Wuhan, we could look at the growth rate in reported cases and get an early estimate that this reproduction number was around 2.4. This is higher than common influenza, which is more in the range of 1.5 to 1.8, but actually much less than some of our childhood diseases, such as measles. This was subsequently then revised upwards in early March as the epidemic started to take off in Europe. We saw faster growth rates, and therefore our best estimate now is the reproduction number is around 3, which is actually very similar to the reproduction number for the initial SARS outbreak back in 2003. The second question then, once we know how fast a virus is spreading, is how severe is infection going to be? And we generally try to characterize this by a quantity known as the infection fatality ratio. That's the proportion of all infections that result in death. And importantly, this denominator needs to include those infections that may be mild or indeed asymptomatic, so not exhibiting any symptoms, because that gives us an idea of how far the virus is spread in the population. And without a test for asymptomatic infection or widespread testing, that's of course very difficult to estimate. However, we had one opportunity in late January to get an estimate of how many people were infected in Wuhan. And this is when those foreigners that were resident in Wuhan started to be repatriated on flights back to their countries. And most of the individuals on those flights, when they arrived back at their home countries, were tested regardless of symptoms. And those very early estimates that were done for an entirely different reason, for public health purposes, to check nobody was infected, gave us an estimate of the prevalence of infection in Wuhan. And through that, by looking at the number of reported deaths and taking into account the delay between becoming infected and developing symptoms and then onward outcomes, we could get an estimate that gave us an early estimate by the middle of February that we first published of around 0.5 to 1% infection fatality ratio. This is a little lower than the Spanish flu back in 1918, which was nearer to 2%, but significantly higher than most influenza outbreaks since then, and therefore was a cause for concern. By the end of February, we could revise this estimate. We were far more robust, and we had the additional data that came from the Diamond Princess cruise ship that you'll remember was moored up outside Japan. And that gave a very strong clue that this estimate was right. It was in a very similar range. And we could also by then see a very strong age dependency with much higher risk of poorer outcomes in those aged 60 and above. And interestingly, this estimate has actually remained quite stable as we've looked at cases across the world to date. So then, given these key parameters, we can do some really quick back of the envelope calculations. So for an R naught of an R number of 2.4, if infections left to spread across the population, we don't do anything and there are no actions taken, then some simple maths will tell us that about 80% of the population should become infected. And with our infection fatality ratio of around 1.1%, that means around 0.8% of the population could die. Of course, this is our worst case scenario. It's not what we expect to happen. Populations will respond, governments will respond, but rather it gives us a metric by which to judge the need to respond and the strength of those responses. So if we take, for example, the UK population of 66 million, that gives us just over 500,000 deaths. Similarly, for a US population of 327 million, it gives us around 2.5, 2.6 million deaths. And these are actually the numbers that you will have heard in early March. They're actually fairly simple calculations that we can do. Of course, as I've said, this is a worst case scenario. And what we really want to know is how effective different interventions could be in reducing the impact. And so that leads me on to mathematical modelling or epidemiological models, which is what we really work on in Imperial. And these models actually rely on the key parameters. So without that early work on the epidemiology, you can't really make valid projections from the models. However, they also allow us to integrate other knowledge that we have. And a key one in terms of transmission is how many contacts people make in an average day and what locations these occur in. For example, they could be in schools, in the workplace, during a commute, or indeed in the household where much transmission of respiratory pathogens generally happens. And those data do exist in many settings from surveys that have been undertaken over many years. So using this information, we could put these into the models and then work out if we reduce transmission in the workplace or we close schools, what's the likely impact to be. And this is what we did in our report that we released in the middle of March. And what that showed was because the speed of spread was really rapid and had already spread quite far in many populations, and because of the severity of the disease, these two things combined meant that we needed by the early March to rapidly reduce contact patterns. And this was primarily to avoid hospitals and in particular intensive care facilities being overwhelmed. We weren't the only group to do this. Many of our colleagues and modelling teams in both the UK, US and elsewhere in the world have reached similar conclusions by this time. So how then does this feed into health policy? Well, firstly, it's important to note there are a global community of epidemiologists. We all know each other, we've worked with each other for many years, and this community was rapidly mobilised with support from the World Health Organization. And so we were in contact with colleagues, for example, from Hong Kong, from Singapore and the US from January, and all of us were discussing the various options and as well as the various responses that local governments were putting into place. How this then feeds into policy, of course, varies from one country to another. But for many high-income countries, there are scientific committees that come together to provide advice to governments. And in the UK, this includes several subcommittees that feed into the Scientific Advisory Group, or SAGE. And these include a modelling subgroup, but also clinical and behavioural science. And so it is really a multifaceted approach to trying to understand the various options. It's important to note that these committees enable open scientific debate. And I think this is really critical because there is a high degree of uncertainty. Scientists shouldn't all agree. They should bring their different perspectives and their different opinions, and then we debate the relative strengths and weaknesses of different scientific conclusions. The committees also need to be flexible, and this is a new and emerging disease, and information is changing and being accrued rapidly. So as new evidence emerges, then advice can and should change to reflect this. And I think a clear example in this pandemic has been the use of face masks. Early on, there was a concern that we really had no evidence to support or to negate their use, but there was a concern that behaviours might change if you wear a mask. You might feel protected and therefore come closer to people or change your contact patterns. There was also a concern about people buying up very personal protective equipment that was needed in the hospital and healthcare settings at that time. But as better evidence has emerged, and we now have much better scientific data to support the use of face masks, the policy changes. So the scientists provide the evidence, the scientific committees synthesize this evidence, but ultimately the decisions that are enacted are made by elected politicians. And I think this was particularly important in the case of lockdowns, because these are such a high impact intervention, which is unprecedented in recent history, and really was therefore not an easy policy decision to make. There was also many other aspects that needed to go into that political decision other than the science that was informing the public health response, and in particular, the economic consequences. And I think this political aspect is reflected in the different decisions that governments have made around the world. And as we have since learned, and are in fact still learning, some responses were better than others. So what works in terms of control of this pandemic? Clearly, it's important first to note and stress that the pandemic is not over, and we won't know for certain until many years' time when we can review all the evidence. And in fact, we're probably still in the very early stages of a virus that's likely to continue to spread globally for many years. But there are some early patterns emerging. First was that many countries with recent experience in similar diseases, particularly in Southeast Asia and South Korea, Hong Kong and Singapore are some examples, reacted very rapidly to contain onward transmission and infection. They implemented extensive contact tracing and localized controls. And this early action, which was built on prior experience and strong public health systems in place, certainly worked. And those countries so far have experienced relatively small numbers of cases. As infection then spread to Europe, I think it was initially undetected. We now know, for example, from genetic data that infection was circulating in early to mid-February. However, at this time, very few cases were being detected in Europe, and the focus of surveillance on those with a travel history from China at that time meant that actually we missed, in hindsight, some of the infection that was going on. So with hindsight, this lack of a broader surveillance system meant that infection had circulated much more extensively, and coupled with the limited testing and contact tracing capacity that was in place at that time, it proved impossible to contain. And so by the time that the epidemic really took off in Italy, and subsequently in Spain, France, and the UK, it was clear that health systems were going to be rapidly overwhelmed. And it was for this reason that lockdowns, whilst blunt and incredibly damaging, as we now know, to the wider economy, were probably the only option available to sufficiently slow the spread of transmission and to avoid the hospitals and particularly intensive care unit capacity being exceeded. Of course, not all European countries initiated broad lockdowns, and Sweden is often cited as an example of why lockdown was not needed. And we're still looking into this, but there are differences. First of all, it's important to note the Swedish population actually did respond, despite it not having a government order to do so. Compared to movements prior to the epidemic, there were drops of around 30% in local movement in Sweden, according to Google mobility data. And that compares to around 80% in the UK. So less of an impact, but still a significant impact. But there are also differences in the density of the populations. Sweden is much more sparse, and even the household patterns in terms of how many people live together and could transmit infection. And these aspects are things we are learning about through this pandemic and finding, for example, multi-generational households are quite important in spread, and these tend to be concentrated in major urban areas. And so whilst what we've learned, therefore, is that the same intervention is not necessarily needed everywhere. And whilst in the UK in mid-March, much like Italy in late February, the lockdown was necessary because we were very close to having healthcare capacity exceeded, in other countries, earlier and localized responses could also contain the spread. And I think this is an important lesson as we emerge from lockdowns, because strong surveillance, rapid isolation of cases and tracing of contacts, and these localized responses based on local public health knowledge are all critical and can allow us to avoid the need for these broader lockdown measures. So finally, I wanted to get to this issue of the challenges with communication and their uncertainty. And whilst we've worked on many epidemics in the past, the scientific community has probably never been under more intense scrutiny than during this pandemic. And coupled with almost the exponential growth in what I would call armchair epidemiologists, even within the broader scientific community, ensuring our messages are heard and understood has been quite challenging. So a positive has been that the profile of science and its role in society has been raised. And this is really helpful for the future, but also through some fantastic science and medical journalism. I think we've got a better understanding in the general population of epidemiology. And I certainly never thought that the R number would become a mainstream concept on television, nor indeed that logarithmic graphs would appear in the daily newspapers. But one of the biggest challenges, and it's always been a challenge, but it's particularly highlighted here, is that everyone wants certainty. And even science cannot give that uncertainty, although science is often thought of as a hard subject and with definite conclusions. And because of that lack of uncertainty, scientists will disagree. And this is a good thing. We need this debate to be able to move forwards. But the media portrayal of this has overall probably not helped us that much. It's fueled the skepticism of science and thus increased the support for alternative theories, many of these based on speculation and misinformation. So to conclude, what we've learned about this new virus is how it spread, what works and doesn't work to control it. We also have encouraging new therapeutics, such as dexamethasone, as well as improved outcomes for those that are hospitalized. So fatality rates should decline in the coming months. But as I mentioned earlier, we've got a long way to go. Cases continue to rise dramatically in many parts of the world, and we are only just beginning to understand the longer-term health consequences of infection that persist for a significant minority of patients. And of course, as we move on, and as the focus of this conference, attention is focusing on one or more new vaccines, and rightly so. And we do have a very encouraging pipeline that is unprecedented. If trials are successful, there could be a new vaccine as early as January next year. That is optimistic. But it does remain uncertain, and therefore we are likely to need to learn to live with this disease. But I remain optimistic. It is possible, and many countries have demonstrated it's possible, to resume daily life and economic activity if we all make the small changes to our behaviours that are necessary. Thank you for listening.
Thank you very much. Thank you very much, Professor Ghani. That was a really great overview, both of epidemiology and of the story so far, as you say, of this virus. So really very interesting. Lots of questions in my mind. One popping up is just picking up on your, I think, really optimistic, conclusion, I suppose, about, lessons learned and good and bad. The use of these bioarchives, these pre-publication sites for a lot of scientific papers. Of course, a lot of companies attending this conference will have, benefited, used, shared, contributed to that. Is that overall a positive, do you think, or are we in danger, as some scientists have told me, that we're of undermining the rigor and the peer review process, and then perhaps falling into the trap of saying, well, that's okay going forward, just put it up there pre-peer review? What's your thought?
Yes, I mean, I think this is an interesting and a new challenge for us. These preprints have been around prior to this pandemic, probably far less used and almost certainly far less read outside of the scientific community. So within the scientific community, we all interpret it as a preprint, not peer-reviewed, and we have the scientific background to really understand whether it is a good piece of research or a weaker piece of research. I think it has been challenging in terms of the wider media interest in preprints. There's certainly been some high-profile examples that have been unhelpful. But on the whole, I think it's a good thing to have that information out there. We need information flow to be rapid, and it is very challenging to get through peer review in this sort of timescales. It typically takes 1 to 2 months to peer review an article. But one of the really big challenges for scientists even is keeping on top of the amount of information that's flowing out in those preprints.
This is it. It's that whole trying to repair the plane in mid-flight sort of analogy, isn't it? So just going back to the point about the difficulty of obviously of modelling data and then of applying it to one's own country population with all those, as you alluded to, structural, cultural differences. Coming out of this, and you made the great point that the same intervention won't be good everywhere. I think in the UK we unfortunately aren't looking amazing. But do you think the policymakers are listening to, many of them hopefully would listen to a talk like yours? I mean, do you think next time this happens, everyone will be slightly better prepared, both obviously knowing what a pandemic feels like in this century, but also in terms of understanding how to hear science, how to interpret numbers? Do you think that governments and policymakers as well are going to do better where they haven't done as well this time, next time?
Yeah, I certainly think so. And I think we can already see that from the experience of countries that had experienced similar outbreaks in recent years. So, as I mentioned, Southeast Asia experienced the SARS outbreak in 2003. Those countries did act very quickly. On the back of that, they had a familiarity with these viruses. So I think that experience helps, and it helps not just at the government understanding of the potential threat, but also in the wider population. Just having lived through this particular pandemic, I'm sure all of us will be much more aware, much more likely to respond early and change our behaviours. And those behavioural changes are the challenge for this type of virus spreading where we don't have pharmaceutical interventions in place, and always going to be challenging because we need to rely on people to change their behaviour.
Absolutely. And to that, I mean, a lot of the companies and investors listening to this will be involved with or know of the many incredible efforts to develop a vaccine, develop a therapy. You alluded to the problem of over-promising, and I think everyone's, here will be aware of that. I wonder what if none of the vaccines do succeed, and we all know most of them won't, that's just drug development, vaccine development. Do you think that the reputation of, do you think faith in vaccine development and preventative measures will be maintained? And just maybe taking on an angle for our investors in the audience, I mean, are you confident that the importance of these kinds of approaches, even if the COVID-19 magic bullet doesn't appear, will be maintained?
I think it's very difficult to say. I think one of the challenges we have with a lot of the alternative theories and misinformation is actually vaccine hesitancy, as we call it. People not wanting to become vaccinated has grown, actually, rather than declined. I think it was starting to see, there was a survey out today in the UK, which showed some quite worrying results about numbers of people who would receive a vaccine, and that's certainly much higher than we've seen previously. So I think those aspects remain a concern. But in terms of confidence, I mean, I'm optimistic that there will be a vaccine. It may take longer, but there are multiple different approaches, and I think that the broad different mechanisms of action of these vaccines will really help us to get somewhere. And as we've already seen, I think the ability to scan compounds for treatment as well has been incredibly helpful. That has grown dramatically in scope, and therefore the ability to set up trials, get products tested, I think will help to increase confidence in therapeutics. One of the challenges, of course, has been not waiting for the randomized trials. And we've seen that certainly with chloroquine. It's important that we have those randomized trials and we have those large international randomized trials for both therapeutics and vaccines so that we get solid answers as early as possible.
Absolutely, and everyone will be looking forward to the vaccine focused panel later on in this conference. And as you say, a wonderful experiment of all these different technologies, almost head to head after the same disease. Thank you so much, Professor Ghani. That was excellent timekeeping, really useful, really interesting commentary. And now I'm going to transition to the next stage of this reflection session. We're going to have a Q&A with Tom Evans, talking of vaccines. He's the CSO at Vaccitech, UK-based biotech company, whose technology is involved in one of the most advanced vaccine programs. So I will say thank you again, and over to Tom. Thank you.
Thank you.